So the first thing we're going to look at here in the central dogma is how we get from DNA to making messenger RNA. And the process by which that is done is called transcription. And the process of transcription takes place inside of the nucleus of a cell, because that is where the DNA is located. And if I want to copy DNA, I got to go where the DNA is to do the job. So transcription is the first process of two processes involved in the manufacture of a polypeptide chain. Again, like I said, it occurs in the nucleus, and it involves a number of different molecules, including the DNA coding strand, the part we call that gene. We need an enzyme called RNA polymerase. RNA polymerase is responsible for making the RNA. We need to have free-floating nucleotides, free-floating A's, U's, G's, and C's that we can join together to make a messenger RNA strand. We need to have ATP to power all of this activity. And we need to have something on the DNA called a promoter region. And this is a region of DNA just ahead of the gene that promotes the binding of RNA polymerase to the DNA to begin synthesizing messenger RNA. And you'll be happy to know that the process of making messenger RNA is somewhat similar to the process that was used to manufacture DNA. So let's go ahead and take a look at this. When we're all done with this process, we're going to have immature messenger RNA, and then I'm going to talk to you a little bit about how we make that into mature messenger RNA. In prokaryotes like bacteria, the steps I'm going to talk to you about right now are all that are needed to make it mature. But in eukaryotes, there's an extra step that I will get to shortly that converts the messenger RNA from being an immature messenger RNA to a mature messenger RNA. Now there's three different stages to transcription. There's something called the initiation phase, which means how we get things started, and that's when RNA polymerase binds to the promoter region. And if you look over here in this picture, here's the promoter region. Well, actually, the promoter region would have been somewhere down in here. RNA polymerase bound to that promoter region and then started moving along the DNA transcript, reading the A's, the C's, the T's, the G's, and bringing in complementary nucleotides on the forming messenger RNA strand. This is the messenger RNA strand that we're making right here. After initiation, we have something called elongation, and then we're going to finish up with a process called termination. This probably sounds similar to some of the things we discussed with relationship to the synthesis of a DNA molecule. So let's go ahead and take a look here oh, I thought I had one more picture in here. I guess I have to do it all on this screen. That's okay. Okay, so we said that during initiation, this is where there's a specific region of DNA that the RNA polymerase binds to. Now one thing to notice, and this is a critical point, you remember that DNA is double-stranded. That means one side of that double strand will be the strand that is the coding strand, or the strand that contains the proper recipe for the manufacture of a protein, while the other strand will be called the non-coding strand. That means the side that is just the complementary piece, but it's not the one that we use to read for the recipe. All right, now once RNA polymerase has bound to the DNA, that's the end of initiation, and we go into the elongation phase. And again, that's when all these free-floating A's, U's, C's, and G's are brought in one by one and paired up with their complementary nucleotide on the DNA, and that allows us to form what you see here in purple, which is our forming messenger RNA strand. That's elongation. Now at some point excuse me, that RNA polymerase is going to reach the end of the gene, and that's where we want to stop copying. There is a way in which the RNA polymerase is told that it has reached the end of the DNA. and that's through something called a stop sequence. There will be a set of 3 nucleotides, and it could be one of several different sets of 3 nucleotides, but that stop sequence basically is a signal to the RNA polymerase that it has reached the end of the gene, and it can now release this new messenger RNA strand from the DNA. Now, if I were a prokaryote, that would be it. I'd be done. Messenger RNA made, and we can move on to the next step, translation. But if I'm a eukaryote, like you and I are, we have an extra step. Remember that everything that just occurred, if I'm a eukaryote, it occurred inside the nucleus of the cell because that's where the DNA is located. Well, that means now I've just made some messenger RNA and it's stuck inside the nucleus. But the nucleus is not the location where I actually make the protein. I have to get that messenger RNA out into the cytoplasm of the cell before I can begin the process of actually reading those codons and making a polypeptide chain. So to get that messenger RNA mature, I have to do some things to the messenger RNA strand to make it so it can get out of the nucleus. And you will remember from your explorations of the nucleus that the nucleus has these things called nuclear pores, or holes in the nucleus. These holes or pores are where different molecules move into and out of the nucleus, and this is where messenger RNA moves through. Before messenger RNA can move through that nuclear pore, 3 things have to happen, and again, these events only occur in eukaryotes because only eukaryotes actually have a nucleus. The first of these 3 events, and by the way, these are called post-transcriptional modifications because they happen right after transcription finishes. The first thing that happens is on the 5' end of the messenger RNA, we add a little cap of nucleotides. Okay, so here's my little 5' cap I just added on to the immature RNA I had right here. The next thing I do is go over on the other end of my messenger RNA strand, and I add a whole bunch of A's, nucleotides, on the other end. Okay, a bunch of them. That's called the 3' poly A, meaning many A's, tail. And here it is. Here's our 3' poly A tail right over here. Last step I need to do before I can get this messenger RNA mature so that it can leave the nucleus. It turns out in eukaryotes, again this is in eukaryotes only, the recipe from DNA is not all in one sequence. Instead, there will be, if this is the DNA up here, there are regions of that gene that are legitimate parts of the recipe for making a protein. Those I'll circle in red. And then between those parts, there's some kind of what I guess you could call it junk DNA. It really isn't necessarily, actually, but for the purposes of our discussion here, we'll think of it kind of like junk. These are pieces of information that are not necessary for the making of a protein. The parts that are necessary for the making of the protein are called exons. The parts that are not necessary for the making of the protein are called introns. So the last thing we need to do here is we got to get rid of all these introns. We don't want a bunch of gobbledygook in there messing up our recipe for the manufacture of a protein. So we have to remove the introns from the messenger RNA. So here's my messenger RNA. It has its 5' cap. It has its 3' poly A tail. You can see in the dark purple it has a number of exons. Those are regions that we want to keep, and they are spread between regions called introns that we want to get rid of. So we're going to use something called a spliceosome, which is a fancy name for an enzyme whose job it is to come in and cut out the introns. So the spliceosome comes in. Here's a couple spliceosomes right here. They cut out the introns. Here are those introns. They are removed. And what's left then, we glue together all of the exons. That's all the good stuff that we want to keep. And now finally we have mature messenger RNA. And now that it finally is all just exons only, it has a 5' cap, it has a 3' poly A tail. Now it can actually exit out through that nuclear pore. The easy way to remember the difference between introns and exons: introns stay in the nucleus, exons exit out of the nucleus. In other words, they are the legitimate pieces of the code that we want to keep. So again, these three post-transcriptional modifications are events that we only see in eukaryotes. They are events that allow us to get that messenger RNA ready to move out of the nucleus. The reason we do not see these events in prokaryotes is because prokaryotes don't have a nucleus, so there's no need to go to these different events to get the messenger RNA prepared for exit out of a nucleus when there's no nucleus to be had. All right, that's the end of transcription. Next up, we're going to see what happens to that mature messenger RNA once it exits the nucleus and the process of translation begins.